Hi, everyone. My name is Etsub Gezahagne, and I'm a rising sophomore at the College of New Jersey. And I'm excited to present what I did this summer in the Kessler lab, working on the effects of Benzoxazinoids on arbuscular mycorrhizal fungi colonization in maize crops. Okay, so before I start, I'd like to introduce with some background information, talking about one of the issues we face as a society, which is agricultural inefficiency and food insecurity. And a lot of this comes down to how we're growing our crops and the metabolism and the ecological interactions that take place in order to grow the crops that we are using today. And so through that, by implementing efficient agricultural practices, we can promote plant soil feedback at the metabolic level to see the results that we want. And so you might be wondering, what's plant soil feedback? Basically, it's essentially when the crops that you're growing in one soil at one point influence the growth and resistance of the crops growing in the following generations in the same plot of soil. And so this, the plant microbiome is very efficient because it allows for the microorganisms that are living in the rhizosphere region of the soil to form these mutualistic relationships with the plant roots so that there's increased resistance and increased growth of the plants. And so one example of this is through the symbiotic relationships that are formed with AMF, Arbuscular mycorrhizal fungi, in which positive plant soil feedback is the outcome, and we have increased nutrients that the plant is able to uptake and then better growth. On the contrary, we also see that plant chemistry plays an impact whereby benzoxazinoids, for example, which are exuded from root soils, from the root of maize soils, influence the colonization activity of the AMF that I'm going to be talking about. And so that kind of begs the question, how can plant metabolism be manipulated to mediate these interactions with root associating mutualists for optimal growth of the crops? And so in our experiment, our overarching experiment was the benzoxazinoid experiment, in which we wanted to figure out how this impacts the AMF colonization. And then we also did a greenhouse experiment in parallel to see the effects of this on live plants, because our first one was an in vitro experiment. And then after that, we also performed a maize root fractionation experiment to see what other kinds of compounds are involved, as well as benzoxazinoids. So for experiment one, the benzoxazinoids are essentially defensive secondary metabolites that are exuded from the roots of maize. And some of their breakdown products include MBOA and BOA, and these are the more stable breakdown products. And in this experiment, we hypothesized that these defensive root compounds reduce the AMF colonization by inhibiting AMF development, such as germination, as well as hyphal elongation through their antifungal properties. And so for the methods of this experiment, we initially had a maize root extract that we created from 2-week-old maize, and then we harvested the tissues to create the extract. We then purified the solution through the rotovap and then created dilutions with methanol at various concentrations that we used. And so for this experiment, like I said, it's an in vitro spore assay and a spore bioassay. And we had 12 replicate plates for every single treatment that we had. And these were at various volumes of the treatment that we added, ranging from 1 microliters to 1,000 microliters. And the treatments that we used were the MBOA, the BOA, the maize, as well as 2 controls, one of them being the methanoic control and the other being just the blank control. And in total, we had 168 plates that we analyzed. So in order to analyze these plates, we looked for the germination by scoring every single plate. And we did this about 6 to 7 days after we had planted the plates. And then we also analyzed the elongation of the hyphae by imaging all of the plates and then stacking them to create a more clear plate picture. And then using a machine learning software called Root Painter, we were able to segment the images to find the hyphae, to do it automatically instead of us having to do it on our own. And so for this experiment, the results showed that the benzoxazinoids did influence pre-symbiotic hyphal elongation. As you can see, there's a decreasing trend in the hyphal elongation as the concentration of the treatments had increased. And this was very eye-opening. So we also saw that the benzoxazinoids do not influence the spore germination, which we thought that they would. And lastly, this experiment also showed us that the maize extract, as you can see, has decreased germination, whereas the MBOA and the BOA treatments did not have decreased germination. And so the takeaways from this experiment would be that benzoxazinoids are indeed inhibitors of AMF presymbiotic elongation, and plant chemistry does affect the AMF colonization that we're seeing. And so this leads us to further experimentation, where we want to know what other compounds in the maize root extract itself, not just the benzoxazinoids, are inhibiting AMF germination. And in order to answer that question, I'm going to first talk about the parallel experiment that we did with the greenhouse, so that we could see from the spore assay what this effect was on live plants. And so for experiment 2, we had this greenhouse experiment where we had wild type maize plants as well as BX1 mutants. So the wild type plants produce benzoxazinoids at a normal level, whereas the BX1 mutants had a knockout of this gene and they produced significantly decreased amounts of benzoxazinoids. So for this experiment, we predicted that the benzoxazinoid mutant plants will experience increased AMF colonization compared to the wild type plants due to the lower levels of benzoxazinoids. And so for this experiment, similar to the first one, we had 14 maize plants per treatment. And so there was the wild type and the BX1 mutants. And we also inoculated them with AMF spores to see how the plant chemistry influences the AMF. And so for this procedure, after growing the plants for about 1 month, we harvested the root tissues and then stained the tissues to then be able to analyze the AMF colonization. And we also prepared root extracts to be able to analyze the chemistry through high performance liquid chromatography. So the results of this experience were very surprising in that we found that the benzoxazinoids did not have an impact on the AMF colonization of either the wild type plants as well as the BX1 mutant plants. And we had expected, like I said, for there to be a significant difference, but there wasn't. And now we know that benzoxazinoids don't have an effect on inhibiting AMF colonization on live plants. And we're not quite sure yet why, but we speculate that it could be because the spore germination might have not been a large enough difference on the live plants to be able to see an effect on colonization itself. So maybe the germination wasn't as strong, but we will continue to do further experimentation with this, maybe by harvesting, instead of 2-week-old maize plants, using 1-month-old maize plants to see if there could be different kinds of compounds that are at play, or even to have a larger sample size. So instead of 14 plants per treatment, having maybe 50 or whatever it could be. And so now going on to the third experiment, we'd like to see what compounds in the maize root exudates are responsible for inhibiting AMF colonization. So this is our experiment 3. And here we did the maize extract fractionation. So essentially we had our maize extract, which we were able to separate the compounds within it by their polarity. And we did solid phase fractionation experiments as well as high performance liquid chromatography. And through this, we created multiple fractions, and we wanted to see if there was a fraction that contains the inhibitor, that fraction would have suppressed AMF germination. So for this one was similar to the first experiment we did. This one was also a spore assay, a bioassay, and we had 9 methanoic concentrations that we initially had, and we boiled them down to 4 concentrations. So these would be 4 fractions that we ended up using. And for this one, the treatments were the 0 fraction, the 2A fraction, 3A fraction, and 5A fraction, as well as a methanoic control and a blank control. And for this experiment, we actually had 6 replicate plates for every single treatment. And so like the first experiment, we scored the germination of the spores about 3 to 4 days after we had planted them. And we also did high full elongation analysis using the software Root Painter. And that's actually an image of when we were training the model, because you have to train the model first in order to recognize the spores and the hyphae so that we could have that be an automated experience. And so for this experiment, the results showed that there was no fraction that we had identified which showed decreased spore germination or high full elongation. And that was also surprising to us, because that could mean that there could be other compounds that we did not fractionate which have an impact. And so we would like to go further with more experimentation here to see what other kinds of fractions would have an impact. And surprisingly, we also saw that there was large variation within the treatments themselves of the high full elongation. So that's something that we would note for future. And to repeat this process, we would like to have an increased sample size as well. So with this experiment, we had 36 plates in total. And so we'd like to maybe double it or triple the size of the plates. And also, we could also use older maize samples. So instead of 2-week-old maize, similarly use 1-month-old maize or even older than that. And so from this experiment, we learned that there wasn't a maize fraction which we identified that contains the inhibitor that we were looking for. But with future experimentation, we would like to see what this inhibitor is and how it's able to, like, what drive its inhibitive properties. And we'd like to learn more about this inhibitor. And so to conclude my presentation, I would like to summarize that we learned how benzoxazinoids influence AMF colonization in maize crops, as we saw through the suppressed hyphal elongation of the spores. And we also learned that there was no inhibitor that we found in the maize extract, which seemed to reduce AMF germination. And so the importance of this research is that for future sakes, we would like to implement agricultural practices that could enhance maize crop yield by suppressing these inhibitory effects of the compounds that the benzoxazinoids, as well as other inhibitory compounds. And I would like to thank Andre Kessler, Ethan Bass, and my lab members, as well as Delaney, Megan, and George, and BTI, NSF, and Cornell. Thank you. Oops. Thank you everyone for listening. Yes, Bree.
Hi. I can't pronounce the name of the compound, but the BXs that you were referring to, are those only in monocot or eudicots?
Well? I'm actually not sure about that, but they are in maize, and they're the defensive secondary metabolites that are exuded there.
Okay. And one more question. For your second experiment, how did you measure the root colonization and the actual roots that you were doing the experiment?
That's a great question. So after we stained the roots and we were able to analyze them on the microscope and take pictures, there was actually a software that my mentor, Ethan, used, which is able to automatically identify the colonization. So we didn't have to manually do that.
Congratulations. I think you had a beautiful presentation, but I just wanted to know, what would be your favorite moment or favorite part of this research?
There's a lot of moments that I really enjoyed, but I really enjoyed working with my lab members and being able to learn so much in such a short period of time. I think that like my lab members and everyone made it very easy for me to learn and to ask questions and to not be afraid to be wrong. So I enjoyed that it was a very welcoming group of people that allowed me to learn. No. Also the field work, I enjoyed that. We would go to the field. We went to the field a lot, but yeah.
So did you check if your maize extracts had benzoxazinoids in them?
Yes, we actually did check that. So we had a lot of extracts that we diluted with methanol, and then we ran HPLC standards. And through that, we were able to analyze the peaks and see that there was benzoxazinoids present in all of them. So that's what allowed us to go further and see which other compounds were present.
But those fractions with benzoxazinoids, did they inhibit the?
No, we did not see inhibition of those.
Not even with the ones with benzoxazinoids.
No.
Okay.
Okay. Yeah.
What's up? I have two questions for you. One, I had three, he took my first one, but my second one is, what made you choose that metabolite specifically? Like, why was that the metabolite of interest?
That was actually chosen before I came here, but so I was excited to just work on it, but there was not that much research about benzoxazinoids effects with mycorrhizae like in corn. So I think it was interesting to see what more we could learn from that and to like get more solid research from that. But I think that's my answer.
Okay. And then I have a third question, but I'm scared that it's going to sound stupid, so I'm not going to ask.
Oh.
One more question.
I can shout. So it's clear that you did a ton of work in a short amount of time. I feel good about that.
Thank you.
I have one question, and it's, what do you think could have caused some of the variation in your third experiment?
Thank you for your question. I'm not sure yet. That's what we're still trying to figure out, but Yeah, I'm not sure, honestly. Yeah, that's why we need to like do more experimentation and hopefully have like more treatments and a larger sample size so we'd be able to figure it out. But it's a very good question. Okay, thank you.